talk about some chemical biology aspects of the drug seed that we generally develop in our laboratory. So I thank Sakuntala madam for giving me an opportunity to give a talk over here at MMTTC. So basically we all know the drug design and discovery. So which mainly focuses. So let me go with a brief introduction. So where we are and how best we can target some diseases through a specific target discovery process. So in this case, so my lab focuses mainly on cancer cell inhibitors role. So where we identify the targets depending upon the collaborators in need or the chemical similarity or what we know now, what we so-called kind of artificial intelligence or computational measures. So we go with the target identification and followed by this slide will give you, so you can see that. So our role mainly is in relation with target identification followed by the lead identification, and so to some molecules of us, so come up to the preclinical development, so which I will discuss now, and followed by some pharmacological assays that we conducted, and some chemical libraries, how best we could able to synthesize and characterize them. So that will be undertaken over in my lab, which I will be going to explain by the students who did those things in detail. Okay, so basically the inhibitors are drugs. So in the right hand side panel, so you can see the disease graph where we could see so many diseases that targets of our biomedicinal chemist especially. So we basically focused mainly on cancer drug development, so which I will be explaining over here. So this slide will give you the outline or progress of my lab, of course, in collaboration with other universities and my lab. Basis. So as I mentioned over here, so the name compound NPB, which targets bad phosphorylation, so which had an application for breast cancer, so which we have a patent PCT, which is patented, and we have an active patent for 13 countries now. So this is actually is in the preclinical stage, and as well as we are going for clinical trials. So likewise, so AMPC, the named compound which that targets triple factor 3. So application is a relation with gastric and lung cancer. So which is also in the same boat. So in parallel, we are working with these two drug candidates that are moving forward. So long back, so DMBO named compound, so that targets heparinase. So in 2010, so we could able to generate a Japanese patent. So in collaboration with Japan lab, where Professor Kazuki Sugara. So that I will explain that later. So that is also now that this molecule is under the market from Merck. So they are using it as an heparinase inhibitor. So again, SIMO or HDF14, so small molecule azaspirine, so we target JAK-STAT pathway, so which has been patented. PCT has been filed in 2016. So all these things, so we summarize. So where my lab, of course, in collaboration. So these are all the work that we are um, in presently we are focusing. So the right hand side, so you can see our work recently got a cover page article from American Chemical Society Omega. So where we could identify lead structures, specifically coumarin and thiouracil conjugates that really inhibiting the SARS-CoV-2 virus. So which is in collaboration with NIV Pune. So which we did, and we could able to demonstrate for the first time that some of the compound which is having a natural structure, so key structure that could able to target the SARS-CoV-2 virus. So with this, so coming to the this work which triggered when I was a research scholar, so where Ramsesekaran wrote to Nature, so in 2004, where about heparan sulfate glycosaminoglycan, which had a primary role in the metastatic process. So we know heparan sulfate glycosaminoglycans cleaved by the heparinase, an enzyme which cleaves the heparan sulfate, so which has a pivotal role in tumor progression. So by degrading the heparan sulfate, and as well as it uh, as a role in demolishing or the architectural sequestration of growth factors and cytokines decrease will happen once the heparan sulfate chain of a tumor cell get cleaved by the heparinase. So heparinase was found to be a better novel drug target, and which has been interesting drug target for a chemist to work for. So in this regard, so that time, so we could able to see a cell press article from Japanese group where they have shown that heparan sulfate, which is having an ionic functional group like carboxylic acid and sulfonic acid and N-acetyl glucosamine disaccharide structure, that could be mimicked by a benzophenone moiety, which is having an ionic group. Okay, so which has been documented.
कोई कुछ होगा तो बता देना मुझे, आवाज लगाएंगे तो।
So somebody can mute. Okay, so basically, so what is the take-home message from that cell-press paper in 2005 was the where the ionic functional group bearing small molecule could act as an heparinase inhibitor, and it inhibited the catalytical activity of heparinase was documented. So with these two references, so I also presented in the upper panel, you can see the heparinase involvement where the tumor cells are invading. So SEM images were captured, and which has been taken over here. So basically for this purpose, when I was a postdoctoral fellow at Sugara lab at Kobe Pharmaceutical University, so what we did was like we screened at a time all the ligands or the small molecules where this could the idea behind the using of surface plasma resonance technology, robotized technique, so which we used because where heparinase targeting compound could bind to the growth factors or cytokines too. So that was the idea behind it. So thereby, so we used a small molecules array or library by covalent linkage with the gold sensor cell. So in that, when we used and we are passing the growth factors or cytokines to the respective ligand library, so we could able to see such a interaction if there. So that has been quantified by using SPR analyzer. So this slide will give you about the vascular endothelial growth factors binding to the respective compounds, so which we named at the latter table. So you can see, so as the DMBO, which has a strong binding affinity of VGF, so when compared to the other structures. So this is the quantification table where it represents at right panel. So where VGF is binding by DMBO with its structure where difluorophenylic moiety has been attached to the oxygen ring that has a cyclospirine structure. So with this, so DMBO, we identified a lead molecule and which we taken for next further studies. So that time, so in this case, the DMBO was used as an inhibitor. So as the panel A says here or represents here, the negative control which has a flow through fraction of a sulfate intact. So positive control refers with the where the substrate was cleaved by the heparinase enzyme. So when we use a DMBO, so you will see the restoration of about 76%. So that has been restored by the in the presence of DMBO. So mean to say that so catalytical activity of DMBO got decreased when there is a DMBO was present, so a sulfate chain which was not degraded by it. So this study clearly explains the in vitro heparinase enzyme inhibitory activity. So, and we have shown in the B and C panel where we the quantified with a different concentration compound, and we see the percentage inhibition of the compound where we use suramin as the positive control. So we know that so the compound is catalytically active, actively inhibiting the heparinase by DMBO in a concentration dependent manner, and in the cell system. So we also measured the heparinase level, and we could find that the DMBO could able to see the high absorbance, so where it represents so the intact heparan sulfate remains in that system. So overall, so what I would like to summarize in this slide is that so very long back we reported in Cancer Letter, and as I mentioned, Japan patent was granted, whose crystal structure have been deposited at UPAC Cambridge Crystallographic Center. And the another assay, another two more assays we did as a functional studies are to know its metastatic potential. So for this case, we used in the right hand side, you can see here. So we used the mouse osteosarcoma cell line, which expresses these glycosaminoglycans. So where Miyasaka sensei provided to Professor Sugara. So that time, so we used I used this compound, and then we injected the element G5 cells to the black mouse via tail vein, so which where the liver metastasis could occur. So upon the injection of before injecting a tumor cells, we used to add up the either heparin or the DMBO. So we could see the restoration or the metastatic nodules relatively was absent at the either heparin or DMBO treated samples. So our mouse. So indicates that these compounds could have a role by showing anti-metastatic activity in mouse lung cancer targeting cells, and followed by they showed catalytically active in vitro or in vivo. Through this model, we have demonstrated that this compound is active against heparinase. So coming to the so that study was when I was a postdoctoral fellow, GSP which took place, and later when I joined to Bangalore University as an assistant professor. So we worked some more on the heparan sulfate targeting enzyme called heparinase. So for this case, we wrote a grant to Israel Lodusky through Israel government. So we fortunately we got it. So I got connected to him because of Sugara, and Israel Lodusky was a known collaborator who gave heparinase enzyme vectors that time to Professor Sugara. So then I came to know, and then we started this work, and in 2000 almost 12, 13 we started it. My student so CP Baburaju started to work on this type of molecule. So triazole thiazole molecule, which is having a phenolic group or iodo group. At first, he used phenolic functionality. So in this case, so we had come up with the report where we identified a novel class of triazoles, thiodiazoles, as a potent inhibitors of human aperonase. So the scheme chemistry is given over here. I know you are all some biological life science background, so let me skip it. So basically, the summary of the in vitro study shows that some of the compound that we have synthesized showed catalytical inhibition by ELISA method and as well as HPLC method also. So aperonase activity we got decreased when we treated a named compound like DTP, which we identified as a lead structure at this work. So in continuation of that, so yeah, so these are all the chemistry part where where some others are reported and which we took as a reference. We also developed a new method. So where we adopted multiple synthetic process or steps where we used esters of ethyl benzoate esters of different compounds. So upon treatment with hydrogen and followed by so many cyclization process, we end up with triazole moiety, which is bearing various functional group in the four and sixth position of such a triazole structures. So with this, so this is the library of compound that we have prepared as the attempt in our case the third series. So after the DMBO and another structure, so which we developed at the in 2018-19 year. So this is the library and this is the NMR and followed by C13 and mass spectra, which we confirmed those molecules integrity, and we have subjected these molecules for inhibition of aperonase activity by colorimetric method. So which we could able to see some of the inhibition when compared to again the small molecule amide 98 that they had, okay, as a positive control. So in this work, so we could able to see 4 CMI or 4 MMI that has a least IC50 value with the 3 and 3.1 microgram per ml model. So this is the HPLC analysis of such all the molecules in the presence and absence of substrate, and as well as aperonase has been used. So where radio labeled sulfate in this say the sulfate labeled material was quantified. So basically, so in vitro and in vivo studies, so using xenograph model where the 41 cancer cells was used, and biopsy mice, the compound treatment has been monitored, where DMSO control and as well as MMI, which have been used, where the size of the tumors got reduced. Okay, so with this, you can see the quantification of such a tumor weight. So DMA when compared to the vehicle, so our compound MMI could able to decrease the tumor weight, tumor burdening take place in the xenograft biopsy mice statistically. So which has been presented. So this work published in Cancer, MDP Cancer, so which I didn't mention over here. So with this, I could, so my lab is focusing on the aperonase inhibitor role. So where some of the diverse structures like 3, 4 new structures were presented, where whose role is in vitro. We in detail we could able to demonstrate those compounds targeting aperonase. So again, so we consider during the corona time, so So development of small molecule as an anti-SARS-CoV-2 agent that targets RDRP as confirmed by integrating what was so the biomolecule that is interacting. So in this case, so during the corona time, I had an opportunity to write to NIV director, so that they could able to take up this project, and we had a collaboration and shipped those compounds, and some good activity has been reported. So so basically, so coronavirus, so I'm giving an overview over here, so which is a positive single-stranded RNA, which induce severe respiratory infection, so that we all know, so that emerged in late 2019 from Wuhan. So basically, we all know about the corona, how the economy and the whole process has been changed, including our lifestyle got somewhat modified because of corona. After corona effect, we could be able to see. So in this slide, so we considered RDRP, RNA dependent RNA polymerase, so NSP12, that is responsible for the RNA replication. So that has been considered as the target for us. So in this case, why we choose the inhibitor like remdesivir, so remdesivir has a nucleobase, and as well as altered ribose structure, and which basically works as an RDRP inhibitor. So remdesivir saved many people. So in the COVID, especially COVID wave 2, that you already know. So we designed some new molecules where such a replacement of because remdesivir should be taken by injection. So our aim was to come up with the oral drug where some by using some naturally occurring kind of like coumarin and some other natural small molecule active component or active structure into an account. So for this case, so the we have made a literature survey where remdesivir, molnupiravir, or some other azividine or another nucleobase bearing structures which have been reported. So, and also suramin, which we also taken into an account. So here, what we did was, you can see in the right-hand side panel. So nucleoside or nucleotide nitrogenous base was considered, where non-nucleoside or nucleotide base was taken. So like coumarin structure have been incorporated with the linker. Okay. So hybrid thiouracil coumarin analogs, HTCA, we abbreviated it. So for that, so this type of analogs which we have prepared in my laboratory, this work was done by my student, PhD student Divakar. Now he is working in a Jubilant Pharma as a chemist. So this is the NMR, and followed by C13 and mass, which we took to know the integrity of the compound and completely characterized those structures. So I will skip that. So coming to the table where we obtained this data from NIV, so we could able to see these compounds antiviral effect on SARS-CoV-2 in Viro CCL81 cell line that has that host cell, so which they used. So in this case, whose values were shown here, ns refers with not significant, and some of the compound you can see here. So PC4 and PC5 and PC6 and PC9. So these compounds, so they have showed anti-SARS-CoV-2 activity. So interestingly, these compounds have been also tested with the coronaviral variants. Like you can see the last column where SARS-CoV-2 Delta B617.2 variant also. So which we have used, which they have used, and whose IC50 has been given over here. So overall, so the study found to be unique and a good template for a new drug development. So this work catches in American Chemical Society Omega Journal as a cover page article. So we have shown by in silico that by comparing the structure of suramin and the compound which we have used 5D here, PC4 and PC5. So you can see here the surface representation of the protein followed by the ligand suramin was shown here, and RTP, which is also taken here, remdesivir triphosphate, so which we also taken for a overlay how these structures is binding into the catalytical or NSP12 or RNA dependent RNA polymerase hydrophobic group, so which we considered, and those interaction have been elaborated here. You can see, so 5D is somewhat unique where the coumarin is binding pattern was different, which we found, and this work has been published, as I said, in ACS Omega. So this is the work. So I will just skip this one. This is the computer model which tells that how these compounds are comparable by computationally with regard to liver toxicity or CYP inhibition, and followed by if there is any blood-brain barrier. So BBB, where you can see the remdesivir triphosphate, so which cannot. So, but 24E, it could cross the blood-brain barrier. So this unique property some of the compounds are showing. So these minute or new information will be helpful to develop a drug. So in conclusion, so I could able to say that we designed a novel hybrid thiouracil coumarin analogs, so as an SARS-CoV-2 viral inhibitors. So further, we also demonstrated that the in vitro RT-qPCR data, so where some of the lead structures could able to show inhibitory effect where it was what to say, they are not toxic to host cells. Thereby, selective index was higher. Okay, so that has been documented and tabulated. So some more information and data you can refer the manuscript which we published 2 years back. So with this, so that is the study which we conducted in relation with SARS-CoV-2 viral inhibitor discovery. So coming to the azaspirins oxygen structure that we reported in almost 2014, we reported this work in 2014 where we identified some of the azaspirin structures. So that could target JAK-STAT pathway in hepatocellular carcinoma. So this work got published in JBC, and we obtained a US patent too academically. So why the study got initiated? Because the naturally occurring these type of structure like starosporin or etripamide or some more naturally occurring complex or bicyclic or tricyclic compounds could able to show biological activity, but their synthesis is very difficult. So in this regard, sorry, in this regard, so we used azaspirine and we prepared the azaspirine structure, the library, and we tested these against hepatocellular carcinoma cells. So this is the table. You can see the raw data which we have presented over here. So where the tested compounds, almost around 20 compounds that we tested. So HDF14 is found to be the the lead structure, which we whose IC50 is found to be 7.3 micromolar. So with that, so when I was a poverty fellow, where the Karnataka University get me the fellowship. So I was at University of Cambridge. So in Cambridge, I could able to demonstrate by learning by myself this artificial intelligence technology, or to say chemoinformatic platform, where there is an integration of target classifier called naive and Parsons-Rosenblatt. So these 1,3 oxygens, when I subjected with the known chemogenic database, the where Andreas Bender group developed it. So in this case, 1,3 oxygens could able to target the target JAK-STAT pathway. So this is the work which I obtained when I was there. So basically it bound to the kinase receptor. So at day 14. So when we obtain the data, so with a good score, you can see here probable primary targets which has been tabulated, and at day 14. So which could able to show its similarity pattern with a value of score of 0.394. So in this case, so in summary, what I would like to tell you is that so the compound anti-cancer activity has been devised in a way like this compound could able to bind to the JAK-STAT receptor and then trigger its action, or its anti-proliferative effect in hepatocellular carcinoma cell. So this slide will give you the structural information where at day 14 NMR and was mass identification. So further, so I will skip that introductory slide. So we have clearly shown that these compounds at tested concentration, phosphostat 3 got inhibited, where the compound could bind to the receptor, tyrosine receptor, and followed by the inhibition of phosphostatic could take place, so which has been demonstrated. And further, some more experiment like dose, time, and as well as translocation static DNA binding, which has been showed here. So where the compound at the 14 could able to decrease the binding of STAT3 to DNA, which has been documented. So further, so you can see here the Western blot analysis of JAK2, phospho-JAK2 got decreases. So phospho-JAK2 and phosphorylation of STAT3. So both the signaling is abolished when the compound treatment was there at HCC. So these are all the functional studies which also explains the cytokine-induced migration and invasion of tumor cells upon subjected with the compound. Naturally, we observed the effect in decreasing such a cell migration and invasion by the compound. So let me skip the in detail in vitro data. So on this one immunohistochemistry data where HDF14 depletes the nuclear localization of STAT3 in HCC cells. So we determined and we examined these studies keeping paraffin as a control. So in summary, we could able to demonstrate this compound is effective in vivo system also, where 2 mg of HDF14 is enough to have a statistical inhibition of tumor size in a node mouse system. So coming to the in silico studies, so and graphical representation of our compound, how it binds. So it may bind to the receptor of tyrosine kinase and followed by the respective phosphorylation of STAT3 and inhibition of heterodimerization of STAT3 could take place, and followed by its anti-cancer effect could be observed. So which has been published in JBC. So further, so we also try to we generated some of the structure like discoumarine. So they are found in nature, and we synthesize almost now Kirti, my student, who she got Commonwealth split side fellowship at Cambridge, the same mentor, Professor Andreas Bender, so where she could able to demonstrate some new structures that could bind to the tumor necrosis factor alpha, which has been documented. So in this regard, BAHC that we named where biscumarin is having a imidazole moiety as an R group, which has been mentioned over here. So the another the molecule, so the crystal structure of a TNF dimer, which has a what to say, the small molecule ligand that has been disrupted the trimerization of TNF, active TNF, which was reported, and we took such a crystal structure and saw and compared with the known ligand which that disrupted the trimerization of TNF to dimer. So where our compound could able to reside at the same site where the co-crystal ligand could able to was occupied. So in summary, what I would like to tell you, so by in silico and as well as by computational method, we could able to show the lead structure resides at the same site or same orientation where the co-crystal ligand of the TNF dimer, which has a hydrophobic tyrosine residues, which are mentioned here. So in cellular system, we also could able to show this compound inhibit the NF kappa B P65 phosphorylation. So mean to say that the TNF binding by the compound. So and then the compound triggers its anti-proliferative effect by targeting NF kappa B. So that has been studied. So when we submitted to JBC, so the reviewer again he questioned to show in vivo model, and we have studied its effect in inflammatory model where we observed colon length and other parameters, so which has been found, and where BASC the compound showed its effect, anti-inflammatory effect in summary. So with this, the journal recognized our work, and it has been become a cover page candidate basically. So at the time, so in summary, what I would like to tell you here is that synthetic biscumarin could have a binding affinity to TNF, so which has been shown using computational model, and then which has an anti-inflammatory effect, which has been documented. So with this, so now I focus almost 3-4 drug targets of our interest. So the another one which I mentioned during the outline of my presentation. So where triple factor 3, which is again one more drug target, which has a very pivotal role in gastric cancer mainly. So our aim was to monomerize the dimeric triple factor 3 by a compound called amino nitrate. So which that inhibited the TFF3 dependent cancer cell survival. So in this regard, so we consider triple factor 3 why, and then the disease breast cancer. So that I will skip this introductory role. I'm sorry. So we will take it up this for graduate students. So mainly, Yeah, this also skip in relation with breast cancer. So ER positive and triple negative breast cancer. So types of one. So in one word, so breast cancer is very important. What to say, disease to be considered to target by a new drug-like compound. So in this case, so breast cancer, so classification in ERPR, hormone receptor, and HER2 receptor. So with a respective percentage has been summarized over here as taking literature. So so in here the current treatment options also have been listed. So you can see. So basically, in spite of these treatments, so breast cancer is the fifth most common reason for all cause mortality in women. So naturally, so in this case, so in relation with breast cancer, so triple factor 3, which is also found to have a role. Those facts have been listed over here, where TFF3 is the driving factor of cancer, which is involved in the proliferation, invasion, so as well as angiogenesis of cancer cells. So and also which is very widely distributed in human body. So we consider TFF3 as the target of our interest. So in this case, let me skip. So some of the examples to show why we choose a triple factor 3 as the target. So, coming to the work where we basically uh, took interest in computationally. So, basically, a triple factor 3 monomerization, which leads stability, that has been proved by from our side from using Cambridge lab, especially Professor Andreas Bender help, where we could see so the dimer structure of a triple factor 3, if we consider. So to in order to understand its structural integrity, where SS bond that got a dimeric form, so which has a flexible region. So if we the inhibitor, if that could able to monomerize the dimeric form of triple factor 3, so that really works against triple factor 3, that which we observed, and we could able to show that monomerization could be possible. Okay, so because high energy biomolecule, so we confirm by some theoretical studies, so and found that the structure could be monomerized by decreasing or by breaking the double S bond, disulfide bond of the triple factor 3 dimer. So which is also in support where we studied molecular biology experiment, so where it is essential. So coming to the ligand binding proposed mechanism, which I proposed for the first time, where triple factor 3 dimer of which is going to be reactive of the nitrile group of AMPC. So thereby, so there is a interaction of ligand and then the dimeric cytokine TFF3, so which forms a complex between the acid source TFF3 dimer, so that is acting against the um, uh, upon adding the sulfur would react to the nitrile group of the AMPC, so the complex could able to occur, and once such a iminium ion is formed, the TFF3 dimer going to be the TFF3 monomer, which is having a ligand. So upon or in the presence of water, so this structure going to be degraded and followed by the AMPC, and then the TFF3 monomerization could occur. So which we proposed the way how the molecule is degrading it. So this is the chemistry how one we could able to prepare these type of amino nitriles synthetically. So for this case, we used 4-hydroxycoumarine and then the alcohol library of benzyl or many different alcohols that we have used, and then the condition was like T3PDMSO in the presence of solvent, and we started it depending upon the reaction condition, and to get a yield we my student in this case Kirti HK, she did the chemistry, and so once the amino nitrile had been synthesized, or some more diverse structures had been prepared by Suzuki coupling reactions in the presence of palladium metal ligand complexes. So later, so we also found that these structure would able to bind to the triple factor 3 by ligand similarity method. So that has the additive role where theoretically or what to say ligand based design is correlating with the identification of triple factor 3 binding molecules, BASC or biscumarins were found to be. So this table giving you the IC50 values of these compounds, AMPC against various tissues or cancer cells like endometrial, mammary, lung, or hepatocellular carcinoma. So in summary, so TFF3 inhibitor has a broad spectrum activity against several cancers. So maybe it is true because of the nitrile group that has a high binding where the sulfur or cysteine residues are present. So so in validating how these AMPC could able to bind to triple factor 3, so surface plasma resonance was performed, and where it has a binding affinity higher binding response unit has been observed over time, so which refers with the first first 60 minutes, so which was found to be association and then a dissociation, where the ligand will be there. So if we used the AMPC as the analyte, so if we increase it, so the response unit goes higher. So mean to say that depending upon the concentration, so the compound binding to triple factor 3 was observed upon immobilizing the protein into the streptavidin sense archive. Okay, so mean to say that so by in vitro we could able to demonstrate the compound binding to triple factor 3, and then in Western blot analysis we could able to show that the dimeric form got inhibited in the presence of AMPC and as well as N-acetylcysteine, so which does the same thing. Okay, so with this, in addition, so molecular docking studies which we perform at Cambridge, so which clearly we have a data where the cysteine SS bond is residing near the ligand. So mean to say that, so the hydrophobic or as well as double cysteine dimeric bond could able to have a near vicinity with the ligand. So in summary of biological studies where molecular biology experiments to show all the proteins in relation with triple factor 3, that upstream and downstream genes we have shown that using different cancer cells, so the other gene product got decreased upon treatment with AMPC. So with this, so we also so that work which we have published in ACS translational oncology. So sorry, I didn't mention that. So ACS translational oncology, which we have published that. Subsequently, we also reported some of the biological studies where AMPC inhibition of triple factor 3 that decreased monolayer cell viability and also increased apoptosis and decreased 3D matricial growth of drug resistant trastuzumab resistant BT474 cells. So without resensitization to the drug. Okay. So mean to say that the drug is effective in resistant carcinoma cells, which we have reported in 2017. Okay. So of course so this work is purely conducted at Professor Peter Lab from Shenzhen, Tsinghua Berkeley Research Institute. So coming to the druggability of the compound, so AMPC, so we have obtained the pharmacokinetic and pharmacodynamic studies. So this study was conducted at Bengaluru. Some company provided it. So where the whole summary is that, so the compound we could use, so it has a druggability. So the parameters, so the overall AMPC could be used as a drug seed. So furthermore, more animal studies and toxicology studies to improve or to know the efficacy of triple factor 3 targeting AMPC compound was made. So these are all the in vivo data which we have reported, and some of the papers which so highlighted over here in Nature Oncogenesis in 2019. So which we have shown that AMPC inhibitor, which potentiates MEK inhibition in lung adenocarcinoma. So in other tissues also, the AMPC compound could able to exert its anti-cancer effect. So finally, so we provided this graphical abstract to ACS journal. So you can see here triple factor 3 dimer, so which intact, so where AMPC interacts and which has a role in NF kappa B or AKT or MAP kinase or in STAT3. So overall, the cancer cell survival got inhibited in the presence of AMPC. AMPC inhibits TFF3 monomerization via AKT and STAT3 pathway in breast cancer cell is documented. So with this, so these are all the facts, so which I have mentioned already. So I will go back. So for the some more work that what we do in our laboratory. So this slide will tell you the patented aminonitriles that we have patented in 13 countries. So recently, last month we got Japan patent too. So, so in another work, so NPB, which we name, so BAD, BCL2 antagonist of cell death. So BAD, which is also is of our interest, where we used the such a biomolecule as the target, and we identified NPB structure that could inhibit the proliferation and induces intrinsic apoptosis through dephosphorylation of BAD in mammary carcinoma cell, which we have reported in PNS. So this slide will give you how the reaction or NPB is generated by petasis reaction by taking various boronic acids and library of salicylic acid and secondary amine as the nucleo as the nucleobase. So which we considered, and then the reaction was run, straightforward reaction, multi-component reaction, which we obtained petasis product, and then we subjected these compound in the same way like what we use Andreas Bender chemogenomic model. So we predicted bad protein as the target. So and also some of the in vitro data and in silico and then interaction study shows that this compound could able to bind to the bad bad phosphorylation and could able to inhibit the phosphorylation of bad by NPB, which has been. So you can see here, so the compound is selling by MCE and some more now including Merck. So NPB is under the R&D scale for R&D purposes. So this molecule is commercially available now. So this work I already explained to you. So and another drug target of our interest was STAT3. So where SN2 domain of STAT3 is considered, which has a phosphorylation site. So for this case, so we from very earlier. So we did the studies in when I was a postdoctoral student at Singapore MIT research center. So in this regard, so which we identified PFP. So kind of pyrrolo pyrrole fused pyridine and pyrimidine structures. So which we have identified as the phosphostatry inhibitor. So this is the graphical abstract directly I am saying here. So where we identified this type of newer structure that target phosphostatry in especially in hepatocellular carcinoma cell. For this case, so we design this type of PFP structures. So where you can see pyrrole type of structure you can see in celecoxib and vitinib or ABT 869. So BCL2 inhibitor also this ABT series. So where you can see the pyrrole and as well as the purine scaffold, which is having which has a inhibitor role against phosphostatry. So for this case, we synthesize a series of urea and urea derivatives of pyridine rings that is having various substitutions. So which we have prepared and then tested for its antiproliferative effect and then followed by phosphostatry inhibition. So basically, this work recognized and which was appeared as a cover page in MedChem.com, an RSC journal. So this in summary, what I would like to tell you that some of the pyridine or pyrrole based structure could be used as an anti-cancer agent. Of course, these structures are like building blocks to so many drugs. Okay, so in this regard, so we could able to synthesize a new structure that could target phosphostatry in hepatocellular carcinoma. So and another interesting drug target is BCL2. So where we also studied the inhibitors synthesis in our laboratory. So one of the study that published in PLOS One very long back. So this work in collaboration with one of my friend now is who is working as a professor in Amity University, Manoj Garg. So who conducted this biological studies where we could able to demonstrate newer aminonitride. Okay, so which has a BCL2 target in acute myeloid AML cells. Okay, so this is the scheme how we prepare these structures by using T3P. So initially T3P DMSO make a complex, and then those complex will react with the amino alcohol moiety and followed by the other reagent additions and the respective finally the respective cyclization occur to obtain the amino nitriles has been documented. So with the synthesis scheme is reported in class 1. So the same way we identified the drug target by chemogenomic method. So our Neobase and Parzan Rosenblatt classifier, which we use in both the cases, the prediction for the 2 amino chromine nitrile that we prepared, so could target BCL2 by in silico method. So which we had a high score for that. So again, the Western blot analysis shows that by using an anti-BCL2 antibody, so initially the expression was found and in many cells, in many ML cells, and then upon compound 4G treatment, you can see the decrease in the BCL2. So amount indicates that the compound could be binding to it. So in another work, so I'm just giving you the recognition which that taken place where the oxygens that is having uh, ether and diphenyl ether structure and then the imidazole chloroimidazole group. So this type of newer structure could able to inhibit the human methynyl tRNA synthetase. So this work is very important because the inhibitor which we identified for the first time to the respective synthetase that has been reported in RSC journal. So coming to the other work, yeah, which I have explained about BCL2. Okay, so yeah, so I explained it. So yeah, so this is the in silico data where BCL2 binding by the compound new the aminonitrile which has no Suzuki coupled product. So here 4G the compound 4G so which has a unique binding when compared to navitoclax. So which has been shown here. So this work got published in PLAS One long back. So with this, I thank you one and all. So